Hello everyone, I roll number 99 and my topic is biosensor. A biosensor is an analytical device containing an immobilized biological material, enzyme, antibody, nucleic acid, hormone, or whole cell, which can be interact with an analyte and produce physical, chemical, or electrical signal that can be measured. An analyte is a compound, example glucose, urea, drug, pesticides, whose concentration has to be measured. As shown in this figure, father of biosensor Leland C. Clark invented the Clark oxygen electrode, a pivotal device that allows real-time monitoring of patient blood oxygen levels and has made surgery safer and more successful for millions around the world.
Good afternoon, everyone. Today we discuss about biosensors. As roll number 99 discuss about the introduction of biosensor, I roll number 40 continue with the main components of biosensor. The main component of biosensor are sensor, transducer, amplifier, processor, and display unit. Sensor. It is sensitive biological element or biological material, example tissue, microorganism, organelles, cell receptor, antibodies, nucleic acid, etc. Transducer. Transducer is a device that converts energy from one form to another form. In biosensors, transducer convert the biochemical activity into electrical energy. Amplifier. An amplifier is an electronic device that increases the voltage, current, or power of a signals. Amplifiers are used in wireless communication and broadcasting and in audio equipment of all kinds. Processor. A processor or microprocessor is a small chip that residue in computers and other electronic devices. Its basic job is the receive input and provide the appropriate output. Display unit. A device with a screen that displays character or graphics representing data in a computer memory. It usually has a keyboard light pen for the input of information or inquiries, such as VDU, Visual Display Unit.
Hello everyone, I am roll number 46 and I will be discussing the working principle of biosensors. Biosensors are based on the principle of signal transduction. Basically, signal transduction known as cell signaling, and cell signaling is a transmission of molecular signal from a cell's exterior to its interior. Signals received by the cell must be transmitted effectively into the cell, which means it is used to ensure an appropriate response. Bioreceptors is allowed to interact with a specific analyte, and the transducer measures the interaction and outputs of a signal. The intensity of the signal output is proportional to the concentration of the analyte. The signal is then amplified and processed by the electronic system. Analyte diffuses from the solution to the surface of the biosensors. Analyte reacts specifically and efficiently with the biological component of the biosensors. The reaction changes the physicochemical properties of the transducer surface, which leads to a change in the optical and electronic properties of transducer surface, and also change in the optical and electronic properties, which is measured and converted into electrical signals and later on detected. Before it, signal is often and amplified. Thank you very much.
Hello everyone, my roll number is 43 and I am discussing about the type of biosensor. Type of biosensor 1. Calorimetric biosensor 2. Potentiometric biosensor 3. Acoustic wave biosensor 4. Amperometric biosensor 5. Optical biosensor. Optical biosensor. Optical biosensor consists of biological equipment, detector element and transducer associated with both component. The biological component is generally a protein target bioreceptor which is immobilized on the surface of the transducer. The OBS are coupled with microfluid which introduce an anacalite as well as separate unbounded molecule and is a powerful biophysical tool for characterization of quantitatively.
Hello everyone, I am Razia, roll number 47. As the roll number 43 explained the introduction of optical biosensor, I am going to explain type of optical biosensor. So the first one is surface plasmon resonance, second is fiber optic biosensor, third is absorbance based biosensor, and fourth one is luminescence based biosensor. Surface plasmon resonance. physical phenomena for which the electrons in thin cold film can excited into collective oscillation. Collective oscillation means to give collective excitation to electron by bombarding it with light of particular frequency. These occurs on the surface of metal at the interface of two media, usually glass and liquid, is illuminated by polarized light at a specific angle. Polarized light is the light waves in which the vibration occur in a single plane. This generates surface plasmon and reduction of the intensity of reflected light at a specific angle known as resonance angle. And this effect is proportionate to the mass on the surface. SPR is used to measure the binding kinetics and affinity of molecular interactions. Now the next one is fiber optic biosensor, analytical device in which fiber optic device serves as transduction element. Transduction element is used to convert non-electrical signal into electrical signal, and it is to produce a signal that is proportional to the concentration of chemical or biochemical to which the biological element reacts. Biological element is proteins, enzymes, cells, tissues, etc. Transmit light on the basis of the principle of total internal reflection. Total internal reflection means complete reflection of a light ray reaching and interfere with a less dense medium when angle of incidence exceeds the critical angle. Based on the transmission of light along with the silica glass fiber or plastic fiber to the site of analysis. Luminescence based biosensor. Luminescence is the light that is not primarily generated from heat. Light output from a biochemical reaction and it can be used in detection of bacteria. Use firefly luciferase from the tails of wild fireflies. Firefly luciferase is the light emitting enzyme responsible for the bioluminescence of fireflies. Bacteria is lysed and yellow light is given off. Luminescence based biosensor used for the detection of metal bioavailability. Absorbance based biosensor. In this, colorimetric test strips is used for color and it measures changes in light absorption. Cellulose pads embedded with enzymes and reagents. Cellulose pads is used to provide maximum cooling effect by absorbing and retaining water in it. And it used in diabetes regulation. Diabetes regulation means how insulin and glucagon regulate blood sugar. Absorbance based biosensor is used to detect particular analyte through color changes.
Hello everyone, my roll number is 1791301048 and I explain about applications of biosensor. Number 1 is food analysis. Number 2, study of biomolecules and their interaction. Number 3, drug development. Number 4, medical diagnosis for both clinical and laboratory use. Number 5, environmental field monitoring. Number 6, quality control. Number 7, industrial process control. Number 8, manufacturing of pharmaceuticals and replacement organs. Thank you.